Hello. All right, I'm recording this because live stream cuts me off and it's ridiculous. And of course, as soon as I hit record, my dog. So we're going to read out of my microbiology book today. So come with me to get my dog and then we'll read microbiology. I guess I can't flip the camera while I'm recording. Apparently, that's like a thing. Yes, Angel, come on. Booger. Now you want out too, don't you? And then you're going to torture the chickens and cause more trouble, aren't you? Why don't you just chill on the deck with me, okay? So this whole virus thing, I just left off a live stream because it kept disconnecting, and I was getting to the part about what your immune system does with a virus, and then I was like, gosh, I don't remember all these details. Let's look at the microbiology book. So anyway, I want to read to you, it's called Microbiology and Introductions. This is just your basic foundational microbiology information on viruses. And, you know, there is some truth to the thing about that viruses not being alive. You know, they can't replicate outside of a host cell, but it's like an infectious kind of a protein that gets carried on dust or mucus or parasites or food or your fingernails or dirt or fecal matter, like dust floating through the air, you know, and it can still get in you and then invade your cells. And when it has a gain of access genetically engineered into it, like the coronavirus. No, I don't match. Look, I bought those when I was a piano teacher. But anyway, when it has gain of access genetically engineered into it, like the coronavirus, I'm going to look like a crazy person. I just don't care. It's not a normal flu, and so you can't be running around. I mean, you can. You have the right. Like, you have the right to be a moron. You have the right to leave your house, and you do have a right to protest. Like, it's in the Constitution, okay? I'm not going to Gestapo on anyone I know or love for protesting, and you have to feed your family, too. And I know people who are protesting because they want to feed their families, and so I'm not trying to discourage people from fighting for their rights and speaking against government tyranny or any of that. However, I believe that the virus is a real threat, and Trump is probably trying to protect people from dying. I do think that. I don't know if he understood the $3.7 million that went to the Wuhan lab under the Obama administration with Anthony Fauci through the NIH and National Institute for Allergy and Infectious Diseases. That was studying bat viruses after it was deemed unsafe here in this country, which is basically going around the law. I don't know if he really understood that, but he's supposedly investigating that now. Meanwhile, people are taking information and like believing what they want to believe to make them feel better, and that's the part that I'm trying to shatter. I'm trying to shatter your reality, your false reality that you've created to make yourself feel better, because you could die from it. I mean, like, so this is not a normal flu. This is genetically engineered from a lab. Let's talk about what even a normal virus is. But, like, I literally just passed the part where it talked about how they grow viruses in the lab. Viruses are different. How are they different from bacteria? So this is chapter 13, viruses, viroids, and prions. Notice that they put prions in the chapter with viruses. Do you know what a prion is? Well, if you're one of those people saying viruses are nothing to worry about because they're just proteins, then you probably haven't heard of a prion, which is like what causes mad cow disease and Kuru disease and horrible things, and it's just a protein. And bleach doesn't even kill that. Proteins can do a lot of damage, okay? Golly, I'm not trying to laugh about it, but viruses, they can be physically seen. It just takes the scanning electron microscope. However, oftentimes it's diagnosed with what's called a Western blot, which I believe I just read here is actually checking for antibodies, which we can do a whole video on antibodies. I'll do a whole video on antibodies for you. Golly, quarantining has made me so fat. Oh my gosh. I've been cooking at home, but let me tell you, I like meat and I like dairy and I like pasta, so shit. Anyway, here's a funny thing called influenza, crossing the species barrier. The irony is that crossing the species barrier is what this vaccine research really is about. I think her name is Xi or whatever, G, I don't know, she's the Chinese lady that worked at the Wuhan lab, was publishing articles either 7 or 10 years ago about the gain of access function and the bat viruses that she's trying to get to infect cross species, that she's physically in the lab trying to get it to do that in the Wuhan lab. Okay? Now, there are 30 strains going around the USA right now, so sure, maybe you'll catch a mild coronavirus. Maybe these people tested positive because they had the flu shot. There's evidence to support that. But there's also evidence to support that at least one of these strains, if not all these mutants, probably not all these mutants because the common cold is a coronavirus, but at least one of these strains is a deadly bioweapon that causes organ damage and lung damage. It can make you throw clots and have a heart attack if it attacks your heart, which has ACE2 receptors. It can take your kidneys, which has ACE2 receptors. It can give you diarrhea. That's why the government bought up all the TP, because your gut has ACE2 receptors. It can tank your lungs. It can make your oxygen levels look really, really low, even though your lungs aren't really that bad, because it's destroying the capillary walls because of the ACE2 receptors and the regulation of blood pressure. That's why people with diabetes are dying from it. It's not the diabetes. It's they have circulatory problems, yes, from the diabetes, but their drugs that they take, the diabetes drugs, affect the ACE2 receptors. So the mainstream media doesn't tell you that. They don't tell you that it's not that they have diabetes necessarily, although the lack of circulation would go with that, but the fucking drug, excuse my language, that they're taking allows that virus to have a super crazy access through the ACE2 receptor, which go watch my last video if you want to know about ACE2 receptors. Anyway, I'm trying to find the part. There are different kinds of viruses: helical, polyhedral, enveloped, complex viruses. The bacteriophage is a fun one that I like to talk about in my old videos because it literally looks like a vaccine. It like injects itself into your cell the way they inject viruses into you, you know. But I don't think this one is a bacteriophage. It is I found it here. Coronavirus is going to be of the viral family Coronaviridae, so it's a viridae. It's associated with upper respiratory tract infections, and it's 80 to 160 nanometers wide. So that's the general class of coronaviruses, the normal, naturally occurring upper respiratory cold coronaviruses that make up, I believe they said somewhere in here, 20% of flus. So those are flus. Those coronaviruses are flus. Do the tests that they give false positives for this? I don't know. I have not researched the test. I am not talking about the tests. I know that some of them had tested positive for coronavirus. Did it test positive for a common coronavirus or the bioweapon version? I don't know. My conspiracy theory brain would like to question that one, but I can't. What I am talking about is the version of coronavirus that was released from the Wuhan lab, which is not a normal flu, and it is a genetically engineered bioweapon. And that's the one I'm trying to get into your denial brains. Okay? I'm trying to get past the cloud of denial. Because it's so hard to, So, growing animal viruses in the laboratory. Let's read, shall we? This is what I used to do videos about. Biotech Today. Go watch it. I just made it public. It's been private for a few years. Go watch it. My very first episode, I show you a website selling human tissues. Right after the Planned Parenthood video thing happened, and I'm just furious about it. And I had way better hair back then, and I wore makeup. It was amazing. I actually gave a crap. Then I, got robbed and put all my stuff private and was homeless for 2 months, and then it was crazy. And now I'm in Florida and I'm recovered, and now there's this. So I would like to share those videos with you. Okay? Growing animal viruses in the lab, which is exactly what I used to just love to always talk about right before my blog got purchased by China. Did you know that one? Did you know that EdenCultures.com? You can see in those videos I would show my website and I would show my health products and I would show my article. You can see the article headlines. During that time when I was robbed, completely distracted, being just in a complete state of heightened sense of threat for 7 months, during that time I lost my blog, and just so happened that China bought it. Go find it. Go look. EdenCultures.com. Maybe you can buy it. They wanted $2,500 the last I checked for my blog, Eden Cultures, and it was a Chinese domain hosting company. So, whatever. Think whatever you want. I'm just giving you facts here. Go look. EdenCultures.com. Maybe you can find some of my old articles on the archive. I don't know, but I lost it all. It's gone. It's all gone. I still have my YouTube videos, so check them out. Check out my Biotech Today series. Seriously. Check it out. So let's talk about growing animal viruses in the lab. 3 methods are commonly used for culturing animal viruses. Living animals, embryonated eggs, which could be a chicken egg, but it can also be a human egg, or cell cultures. If it's in a living animal, okay, this is why I'm just like, anyway. Mice, rabbits, and guinea pigs, okay? Most experiments to study the immune system's response to viral infections must also be performed in virally infected live animals, because there's a lot of what the information about viruses and not being alive and the exosomes are just the immune system's response to the viruses. That stuff's kind of true, but that doesn't mean that viruses aren't contagious, okay? So they are often studying the immune system's response to this virus that they're putting in, which is a toxin, which is like a type of toxin. But if you go and you read what I read about by Dr. Hulda Clark, The Cure for All Diseases, in which she postulates that toxins and parasites are the only 2 causes for diseases. And she talks about viruses as basically being like yeasts are like a small parasite, and yeasts are like a single-celled parasite, basically. Viruses are carried by yeasts and parasites. They're carried, it's like the protein, toxin, or poison that parasite or yeast produces in you, okay? So there's this information about radiation pneumonitis and the exosomes that come out, which is your immune system's response to the radiation pneumonitis, which I'm sure is very bad and does exist. Remember, believing that this is a bioweapon does not exclude you from believing 5G is bad. It's not an either or choice. That's an Illuminati mind control tactic. It's 5G or it's a virus. No, bitch. It's both. There's 5G and it's bad, but there's also a fucking virus. Wake the fuck up. Okay? They want to take your rights and there's also a fucking virus. It's not either or. It's like, here's the guy on top and he's holding the strings and he's showing you the 2 things. It's both. It's not one or the other. You don't have to believe one or the other. You're in cognitive dissonance. You're in denial. Get with it. Start growing your garden. Okay, start collecting freaking rainwater. Get some freaking chickens. Sit your butt at home. It's not going to go away forever. It's going to come back. It's in our environment forever. You have been assaulted. The world has been assaulted. And it's by mad scientists, the same people that I always would make videos about. The mentally ill person that robbed me said, "What did you do to piss off the agency?" I don't know why I said that. He didn't know either. I don't know, maybe he was just mentally ill. But I was making videos about this. I was making videos about this, about the 21st Century Cures Act, which has laws in there that says you can be tested on without your consent. Any injection or test that you get, if they're doing a research thing, they can include you in that research and not tell you. So whatever these tests are, they can include you in some kind of research and not tell you. They were talking about human cell tissue products. Please go watch my video, Mark of the Beast and the 21st Century Chip Act. Because since that, I didn't even know what 5G was, but I talk about wireless devices being charged, nanochips being charged by wireless devices. I talked about that in that video, but I didn't even know that Bill Gates is literally inventing one. I didn't know it at the time. It's crazy, isn't it? It's just crazy. So they infect this animal with this, okay? So it's connected to parasites and yeast are just single-celled parasites. So there's still an infectious agent to it. Do you understand? So the guys that are like, well, they're just showing you antibodies. Well, it's true. Antibodies don't equal immunity. They can trigger immunity if you have an immune system to trigger, but they don't equal immunity. And sometimes they'll check your antibodies and say you're positive for a virus. And other times they'll check your antibodies and they'll say that you're immune to a virus. But really, that's just all bullshit. It's all bullshit. Antibodies only prove that you were exposed to something that triggered your body to make that protein. That's all it proves. It's not the virus itself, okay? But that doesn't mean viruses aren't contagious, okay? So yes, they torture animals to make your vaccines and your bioweapons that you're currently locked down from, okay? So anyway, animal inoculation may be used as a diagnostic procedure for identifying and isolating a virus from a specimen after they're inoculated with the specimen, so it is injected. So yes, that's a really good way to bypass the innate immune system. Our innate immune system is top notch. We need to really maximize that, okay? They are observed for signs of disease or is killed so that infected tissues can be examined. So that's what they're doing to animals. Some human viruses cannot be grown in animals, or can be grown but don't cause disease. The lack of natural animal model for AIDS has really slowed our understanding, they say. They're really upset that they can't study AIDS more because it doesn't cross species and infect cross species. That bothers them. Did you know that there's HIV proteins on this virus? But here it is, this book that they wrote. When did they write this? Let's just see. What year this freaking published? Probably 2010. That's when I was in college. They were mad back then. They were like, oh, we can't really research AIDS enough because we can't infect different animals with it. Damn it. We're going to have to genetically engineer a cross-species virus so we can infect more animals and kill them and look at their tissues under a microscope. This is what they're doing, you guys. And you're now a guinea pig. And some fucking Koch brother idiot is infiltrating the truth media movement and getting you to go out and protest without a mask because they want you to die. Soros? Koch? Huh? It's not a choice. You don't have to choose the Koch brothers or Soros. You don't have to choose either one. You don't have to choose getting infected or immune to the virus or the vaccine. You can just fucking stay home and not get infected with the virus. But eventually you may get infected with it, but it's mutating very quickly. And every time it passes through someone's cell and hijacks their DNA process, it leaves a part of itself behind and it picks that person up. That person's immune system changes it fundamentally too. Think about all the hard times you've been through and how it fundamentally changed you, and you're stronger for it or different forever. It's the same thing that happens to these RNA viruses. They get changed forever. So just postpone as long as you can. It will wither out. Their sorcery is weak. Obviously, otherwise, but I do think that it was intentionally engineered to spread slowly and kill slowly. That's why the lockdown is going along with it. But the problem is that if you don't want it and you don't want to be culled, you don't want to be sifted for weakness with their genetically engineered eugenic virus, then you do need to sit at home. You need to stay home and try to avoid it. Homestead, right? Supplies, like gardening. You know what I mean? Like, if you're worried about 5G, get a radiation detector with your Trump money. Or, install Ethernet in your house and learn about making your own router with a Raspberry Pi and do those things. You can do those things. But viruses are contagious. It's both. I'm sorry. It's like so hard for you to understand. It's like when I walked in and my house was ransacked, and I was too scared to even give the name to the cops because I knew he'd kill me. It's like, you really want to live in denial, right? You really want to live in that denial. No, it couldn't have been. But then there's, he'll kill me. It wasn't him. It's not a choice between he'll kill me and it wasn't him. It's not a choice between getting the virus and getting the vaccine. It's not a choice between the virus is real and 5G is real. It's not a choice between the virus is jacked up and they're trying to take your freedoms. It's all of it. And you don't have to partake in any of it. You don't have to choose any of it. You can stand up and protect your damn self. You can do that. But you got to start by tending your own garden. You got to start by doing that and protecting your fortress, your home. And the people that I mourn for the most right now are the people who are homeless or packed into tiny apartments. Those are the people that I feel for because they don't have the choice. Or the people that are looking for jobs, of course, those people. It's a real problem. I talked to a hairdresser the other day. And I was like, look, I'm just being honest with you. I support your right to feed your family, but I'm not going to be going to a hairdresser anytime soon, especially one that thinks the virus is a hoax. Why would I go to that hairdresser? I'm not going to go there. You might want to find a different way to earn a freaking living. Meanwhile, I'm all fat, but whatever. I can't stand it. It's just how fat I look. Whatever. Point being, guys, let's keep reading about their crazy science. I want you to understand their crazy science, okay? So they're upset they can't study AIDS because it doesn't infect cross species. But remember, this was 10 years ago, so they've taken care of that now. Chimpanzees can be infected with one subspecies of HIV because they don't show symptoms of disease, but because they don't show symptoms of disease, they can't be used to study the effects of viral growth. AIDS vaccines are being tested in humans, but the disease progresses so slowly it can just take years. So they're just complaining about their shit. And then it talks about embryonated eggs and how they take the egg and they can inject the virus. Of course, they can do the same thing to give someone 3 parents instead of 2. and inject certain Dr. Savos, Savos, who's associated with the cult of the Raelians, who has a fertility clinic, I think, in Tennessee, but he also works with Clonade overseas, who creates clones, uses a very similar technology to what I just showed you. Except instead of viruses and chicken eggs, it's human eggs and sperm and cloning. Taking actually DNA from one egg and putting it into another egg. So you could have like two moms and no fathers. Or you still have to have the Y information though. So I don't know. I got to go back and watch that. But it's crazy stuff what they're doing. So then, of course, there's the cell cultures, which is another way that they grow viruses, yeah? So they replace embryonic eggs as a type of growth medium for many viruses. Cell cultures consist of cultures grown in culture media in the laboratory. Well, what is the culture media? Because these cultures are generally rather homogeneous collections of cells and can be propagated and handled much like bacterial cultures, they are more convenient to work with than whole animals or embryonated eggs. Cell culture lines are started by treating a slice of animal tissue or human embryonic tissue with enzymes that separate the individual cells. These cells are suspended in a solution that provides osmotic pressure, nutrients, and growth factors needed for them to grow. Normal cells tend to adhere to glass or plastic containers and reproduce to form a monolayer. And this is a monolayer here. That's what normal cells will do. They'll make a monolayer. Viruses infecting this monolayer cause the cells in the monolayer to deteriorate. So this is where this is an example of not showing you the virus, but the effect of the virus, okay? So they're putting something in that petri dish and it's causing this to the cells, which is not something that we're going to encounter in real life, but it is, if it does get into our bloodstream, this will help us understand what it's doing to our cells. These people that are sick, somehow it got into their bloodstream. Maybe it was from the flu shot. Maybe they ate some poo and bit their fingernails after they didn't wash their hands, after they took a dump. Who knows? I am not saying. I am telling you what it does once it's in there, okay? Right now. But I'm also telling you you can inhale it because it's just reality. Sorry. That is called the cytopathic effect when it deteriorates the cells. Viruses may be grown in primary or continuous cell lines. Primary cell lines are made from tissue slices, but they die out after a few generations. Certain cell lines, called diploid cell lines, developed from human embryos, can be maintained for 100 generations and are widely used for culturing viruses that require a human host. In other words, if they want to culture and grow a virus that spreads in humans, then they use these embryonic cell lines because they are what's called, what is it called? They're called, it tells you right here, continuous cell lines because they can just keep going, okay? So they used it for the rabies vaccine. They talk about that. When they're routinely grown, when viruses are, this is literally what the book says, okay? Just listen. When viruses are routinely grown in a laboratory, continuous cell lines are used. When they're growing viruses, they're using continuous cell lines. If they're viruses that infect humans, they're using human fetal cells. So, sorry, it's just the truth. They are transformed cancerous cells. Here's where it says they are transformed cancerous. Okay? That can be maintained through an indefinite number of generations, and they're called immortal cell lines. One of them is HeLa, which is a woman named Henrietta Lacks. It's her, she died in 1951. They're claiming that it's lost all characteristics of the individual cell because it's been replicated so much. That's a good excuse, but my Messiah tells me that bad fruit comes from a bad tree. If you have a bad tree, it's bad fruit. But oh, my neck, I can't look down and read books. So viral identification, they talk about viral identification, they talk about viral multiplication. So it does admit that it's not easy to identify a virus. They cannot be seen at all unless you have an electron microscope. It doesn't mean they've never been isolated or seen. That is a rumor going around. But a scanning electron microscope, which is can show teeny nanometer size. Remember I told you the size of coronavirus is a nanometer? That can see a virus because it can zoom in literally that far. And nano is like, see, milli is 1,000. Nano is like to the -6 or to the -9. It's very small. Very small. In these tests, they're detected and identified by antibodies. And I just could get into antibodies, but I already railed on antibodies a few minutes ago, so remember what I said. Okay, so that's not... So there's excuse me, I skipped a sentence. There's electron microscope, and then the other alternative is the Western blot, which is where they look at the antibodies, which I said earlier. So they talk about viruses replicating. For a virus to multiply, it must invade a host cell and take over the host's metabolic machinery, which is what I was telling you. A single virion can give rise to several or even thousands of similar viruses in a single cell. So it's not to the power of 2, it's to the power of. This process can change the host cell and usually causes death. In few viral infections, cells survive and continue to produce viruses indefinitely. That would be a question for coronavirus, this particular COVID-19. Although the means by which a virus enters and exits a host cell may vary, because it's different receptors, in this case the ACE2 receptor, okay? The basic mechanism of viral multiplication is similar for all. Bacteriophages multiply by 2 mechanisms. We're not talking about a bacteriophage though, so we'll skip that. So it goes into bacteriophages, and then somewhere in here, I think it said go to chapter 24. Biosynthesis of DNA viruses. But these are RNA viruses, so we'll talk about that later. Interesting information, I don't know. So I'm going to go to chapter 24, which had something else in it here, I think. And then... Infections from animal reservoirs transmitted by direct contact. I wonder if that could apply to this. A normal bat virus, for example, which you're not even supposed to touch the freaking bats if you follow the Torah, let alone a lab-derived gain of access S-spike protein added HIV spliced virus. Just a freaking bat you're not supposed to touch because you can spread it. But you think this is just a flu? Oh, they're fucking nuts. So there it is. Anthrax is one. But these are mainly talking about bacteria. These are bacterial. Viral disease, chikungunya virus by mosquitoes. That is something that we need to consider. These are all things that you can get infected by contact. Lyme disease, Chagas malaria. Those are parasitic related, even though they're not actually, they're calling it protozoa. You can call it a protozoa, but basically that's a cellular parasite instead of a helminthic parasite, but it's a type of, they're a type of parasites. They just don't want to admit it because they don't want to help people to heal by killing parasites. So I'm looking for chapter 24. This is chapter 23. Microbial diseases of the respiratory system. That's chapter 24. And then I'm going to run this off. See if there's anything in here that was relevant. Laryngitis, goodness gracious, DTaP. Viral diseases of the upper respiratory system. So number of viruses are the common cold, 50% are rhinoviruses. Coronaviruses are about 15 to 20%. So I said flu, excuse me, it's cold. 10% of all colds are caused by one of several other, and 40% no causative agent can be identified. So that would be those cases where it's toxins or an energy thing, or maybe you were eating too much junk, or maybe you were run down. But there are viruses that have been identified in the other 60% of the cases. And just because you didn't identify in those cases doesn't mean it's not a virus. They just didn't find it. So we can accumulate immunities against cold viruses, which is why they say here older people tend to get fewer colds. The fact that older people are dying proves that this is a new virus, because what happens is the younger you are, you notice how young kids really survive? They have a very big thymus gland, and the thymus gland is part of the immune system and helps you make these white blood cells. And so when you're exposed to something as a kid, that's really better than getting exposed for the first time as an adult. That's why it's a harder time for people to fight off measles if they get exposed as an adult, or chickenpox, or mumps, or rubella, or whatever. Because our thymus glands are, when we hit, I think, puberty, it shrinks back down. So these older people, not only are they older and they have like comorbid, conditions, and they're on these medications that allow faster access, and they're getting flu shots in the nursing homes, but they also don't have this thymus gland that these young kids have. And so the young kids can definitely carry it, but they're not necessarily going to die from it. So the fact that they're saying this, that shows you this is a new virus, okay? Immunity is based on a ratio of IgA antibodies to single serotypes and has a reasonably high short-term effectiveness. So this plasma shit is short-term, okay? I mean, it might help if you're dealing with it, but it's not like we can just get a plasma infusion and be good for the rest of our life. That's not going to be realistic, okay? But if you're sick, I mean, it could help. I don't know. There are 113 types of rhinovirus alone. A vaccine would not be practical. So that's true. So in here, they do actually talk about how, this is interesting. This is the part I want to include because I'm not trying to be biased. They're saying they're arguing over how the cold is spread. So let's read, okay? They thrive, let's see, we're not talking about rhinoviruses, so I won't talk about how they thrive. Let's see. A single rhinovirus deposited on nasal mucosa is enough to cause a cold. However, surprising agreement on how it's transmitted to a site in the nose. So how does it get to the nose? So they know that having it in your nose, you have the cold. So when they test you for whatever, that's considered having it. Okay, so they're finding the actual virus in you, not the antibody when they're swabbing. The antibody would be a blood test, okay? Because your antibody is made in your blood, okay? So anyway, they agree that it just has to get to your nose, but they don't agree on how. And I already gave you some ways how, but they did do some weird studies here. Let's see. Guinea pigs and the flu virus show virus tend to be carried on airborne droplets of water vapor. And in the air, in dry air typical of low temps, the droplets are smaller and remain airborne longer. So that could be why the humidity helps to filter this out of the air, because the drier, smaller particles float around longer than the bigger droplets. So yay, I'm in Florida. That facilitates the person-to-person transmission when you're in a drier climate and tiny dust particles in the air, not so much water filtering the air, then you're going to have a higher rate of airborne transmission. At the same time, the cooler air causes the cilia of the cilia escalator to work more slowly. So when you're exposed to cold, these little tiny hairs don't work as well at getting it out. Okay, so the hot weather does help with that as well. So here's some interesting experiments with card players that they talk about. So there's a series of experiments involving a group of card players. Half had colds and half did not have colds. Supported the hypothesis of airborne transmission as opposed to other types of transmission. Half of the healthy players were restrained, so they could not use their hands to transfer through their noses viruses picked up from the playing cards. Okay, so the playing cards were contaminated. Think groceries. But in this case, you're playing cards. The cards are contaminated. The dealer has the virus. He sneezed all over it. Okay, just imagine that. Half of the people are restrained. They cannot touch their hands and face. Maybe they're wearing gloves, and the gloves remind them not to touch their face. Maybe they're wearing a mask. Okay, whatever reason, they can't touch their face. The players who could not touch their noses came down with just as many colds as those who could touch their noses. So it didn't help them to not touch their face, okay? An argument for airborne transmission. So let's take the mask out of this equation. Let's just say they couldn't touch their face, okay? So this is making an argument for airborne transmission. Of course, we're talking about colds and flus here, but that's what a coronavirus is. Even when healthy participants in card games were placed in a room separate from anyone suffering from the cold, they're isolated from airborne secretions, but they handled the cards literally soaked in nasal secretions. None developed colds. Hallelujah. I have not been able to disinfect my groceries in like 2 weeks because I literally ran out of 7th generation wipes, and for a while I was like really low on paper towels, so usually I'll use like a vinegar spray with essential oils, or I'll use like 7th generation wipes that have thymol in them, which is like really potent disinfectant. Anyway, I haven't been doing it, but I'm still not sick, praise Yah. So that explains this, because what happens is as soon as it hits that surface, things start to kill it. Sunlight kills it, humidity kills it, heat kills it, isopropyl alcohol, like they talked about last night on the press conference. But even just air, humid air, and 75 degrees and up can kill it. So once it's on those cards, by the time these people got the cards, they didn't even get it. Hallelujah. So that helps me with my grocery stress. But the people that were in there that didn't touch their face versus touching their face, those people all got the same amount of colds. So if you're in the store and you don't touch your face, it really doesn't freaking matter. I think the sneeze guards are probably helpful because if someone directly sneezes, you know what I mean? But it's still going to probably float around. So only if they really have that N95 mask, which filters out those really nano particles, because this virus is nano-sized, remember? So they need that mask that has nano-sized filtering. The problem with that is it's filtering so much that you can't freaking breathe, and it's horrible. And I felt like Darth Vader when I went to the store and I had a mask and goggles on like a crazy person, and I felt like Darth Vader. So anyways, yeah, I have a lot of those all around. So if you're checking that out, just so I have plenty of those all over. Be checking out my system here. So anyway, so let's go back to the experiment with the spread of the airborne virus. So even when healthy participants in card games were placed in a room separate but handled the cards soaked in them, none developed colds. In perhaps less repellent series of experiments, researchers required healthy volunteers to kiss cold sufferers for 60 to 90 seconds. Only 8% of them came down with colds, interestingly enough. Maybe because when they kissed them, there were also probably antibodies in there, who knows, or immune secretions that go along with it. I don't know. I don't know why that is. So, of course, it talks about antibiotics are no use and blah, blah, blah. So anyway, I wanted to share that with you because that was interesting. And I'm going to sign off now because it's been a very long video. And I'm going to just give these links to people when they try to say stuff. So airborne, because it's true, like when Trump said last night, well, if their hand is in the sun, then wouldn't it kill it on their hand, even if they touched the playground where the sun wasn't hit and got the virus? And so it seems like, according to my microbiology book, yeah, it can. So it's not necessarily about touching your face, because the sun and the air could kill it, but it's going to go through the air. So it seems to me like the N95 mask and the goggles, unfortunately, are the way to go. Or getting delivery, which is what I've been doing, is trying to get a lot of delivery. And when I did go out, I wore the mask and goggles. But it's not such a worry about reusing them, because remember, you can just sit them in the sun or in, like, next to your steamer basket when you're cooking, and the virus will die on them. So we should be reusing them, seems like. And so if that, from the information last night, I'm going to add to that and say, it seems like we can reuse the PPE, but the PPE is important. So peace out. Viruses are real. Okay, bye.